Hello and welcome to the University of Calgary webinar, Nanomedicine, the new way to detect and treat cancer. My name is Jennifer Alford and I will be your moderator today, and we're going to meet our scholar in just a minute, but first I want to give you a little information about what we're doing today and how it all works. Now, these webinars feature a series of University of Calgary scholars who bring us a summary of their research. We invite you to ask questions throughout the webinar. Just type it in that little box to the left of the slides, and we hope to get to as many of your questions as we can. Now, we also have some resources you might be interested in checking out, and you can find them in two different places on your screen. To the right, you'll see a box that says Resource List, and down below there are icons, and the resource icon looks like a little piece of white paper on a green background. As we go, please keep the conversation going on Twitter with the hashtag #ExploreUCalgary, and at the end of the webinar, we hope you'll take a minute to answer one very simple, quick feedback survey question. You can find that, again, on the right of your screen, underneath the resource list, or at the bottom of your screen, the icon looks like a little white clipboard on blue. Now, without further ado, to our scholar. David Cramb is a professor in UCalgary's Faculty of Science, and he's the head of the Department of Chemistry. And we just want to make a note here that he is indeed a chemist, not an oncologist. So he can't answer very specific questions about different specific questions about cancer. He's going to talk about the chemistry behind everything. And before we get him to tell us everything he knows about nanomedicine, we have a poll question because we want to find out why you out there in cyberspace are interested in this topic. So you should be able to see those answers there. Why are you interested in this topic? You have suffered from or are suffering from cancer. You have a family member or a friend who has suffered from or is suffering from cancer. Maybe you work in the healthcare system. Maybe you're a cancer researcher, or maybe you're just interested in the topic. I suspect we're going to have a big showing of number two, lots of people involved with cancer through friends and families. Lots and lots of cancer out there, unfortunately. So we'll give everybody a minute to click their appropriate little box there. Well, there you go. Yeah, 31% know someone, and also just 45% of people interested in the topic. I guess, David, because it is such a prevalent disease.
Yeah, absolutely. I think everybody has some knowledge, direct or otherwise, of cancer. I know it's touched my family personally, so it's kind of I feel like a knight in shining armor trying to discover more about cancer and more about how chemistry can help treat and diagnose cancer. So let's start with the story then.
Mm-hmm.
So just to give you a little hint as to what's going to happen over the next little bit. So what I'll try and get across to everybody out there in cyberspace is how we can make connections between the disease state of cancer and nanoscience, which is more kind of a chemistry and a materials science. And it's an interesting story in that what we started off trying to discover led to something else. And that's often the way it goes with science, that you're looking at one thing and then you actually notice something else really interesting happening, and that leads to a potential breakthrough.
Oh, I'm interested to hear how that one went.
So before we get going into the depths of understanding how nanoscience and nanomaterials can be applied to cancer, just a little bit of a background on my understanding of cancer. And so this is maybe the way through a chemist's viewpoint of cancer developing. So in this panel on the far left-hand side, you can see what are normal cells. These are the white kind of rectangles with little dots in the middle. Those are the nuclei of the cells. And these cells are supported by a life-giving system, which will have blood vessels and lymph nodes and that sort of stuff. So this could be, for example, the lining of a person's lung, where the air would be the yellow part above this. And then something happens to some of the cells, and it's usually driven by some strange genetic transformation of the cells, and they no longer can resist dividing uncontrollably. And that leads you to the third slide, where we have abnormal cells multiply. And a cancer can only get, tumor can only get so big under those conditions, and then it has to recruit more nutrients as it grows in size. And that's what the final slide here is meant to be showing, that a cancer's tumors somehow started to develop in its physiology and anatomy. Here we've got something called angiogenesis, and that's the fancy word for formation of new blood vessels. And those can serve as transportation both for nutrients to the tumor and then possibly as tumor cells leaving the tumor and circulating around. And that's where metastases kicks in. So that's kind of the high-level background of a typical cancer of a soft tissue. Something else that we're understanding about the tumor as it grows too is the matrix that it builds around itself. So it doesn't just recruit new blood vessels, and that's what this image is trying to convey to you here. On the left-hand side, we have some stuff coming, what appears to be coming out of the tumor, and that's the proteins that allow it to communicate with the blood vessel system and basically say, hey, new blood vessels, come over here and support me.
So it attracts the blood vessels, the proteins do?
Yeah, and it's kind of a monstrous thing that it does in order to grow further and survive better. It also builds a protective shell around itself, a kind of fortress of a molecule protein known as collagen. And that's kind of like a jello, if you want to think of it as a material. So it builds up this surrounding gelatinous material, which also makes it hard for things to get into the tumor. And that may be a limitation in the kind of chemotherapies that we have right now.
Because the drugs can't penetrate the collagen sometimes?
Yeah, it's like going through a more viscous medium kind of thing. So if you put a salt solution on top of some jello, it'd be a long time before the salt gets to the bottom of jello. That's that sort of thing. So given this evolving understanding of what a tumor is like, also important to get an understanding and background of what we think of when we think of nano, and that's probably something that people have less familiarity with than a tumor. So from a chemist's point of view, we make nanomaterials by assembling atoms and molecules together. So the blue and yellow dots are meant to represent atoms or molecules. Chemists will design those so that when you mix them together, they come together in a specific kind of way, assemble, and then potentially rearrange into a material that has new and interesting properties compared to the building blocks that it was made from.
Now you make nanos, but it appears naturally as well, right?
Yeah, absolutely. Yeah. So this kind of length scale, the 1 to 500 nanometers in dimension, is similar to things found in nature, like collagen, for example. Individual collagen molecules, proteins are about this size scale as well. And so this gives you a kind of a scale for this. So a couple of analogies that might help you imagine how small nano is. That's always a challenge for folks. So one way to think about it is if you imagined a tennis ball or a soccer ball and how big that is compared to the Earth. That's the same difference in size as a nanoparticle on the soccer ball. So huge orders of magnitude. To the soccer ball, the Earth is flat. And I think recently to Shaquille O'Neal, the Earth is also flat. And so so a nanoparticle on a soccer ball, the soccer ball would appear flat as well.
Wow.
The scale that we have here, just FYI, each marker in the scale jumps down by a factor of 10. So it's not a linear relationship here. And you can see that on this scale, the nanoparticles, which are kind of in the 10 to the power of 2, so 10 to 100, in that region of antibodies, so that's proteins that your immune system uses, and viruses kind of in that size range. So considerably smaller than a cancer cell itself and way smaller than a tumor. Right, so here's what folks have been thinking about with respect to nanomedicine and how nanomaterials can help with nanomedicine. And this thought process has been happening probably for 30 years now, maybe even a little bit more than that. So there's a little animation here. You'll notice, so at the top right-hand part of the figure, there is a tumor. There's all these new blood vessels that are growing towards it. The tumor is shooting out these proteins, recruiting more blood vessels. And in the part where the blood vessel is branching off in a couple places, you'll see a couple of black dots on the top blood vessel wall. And those exist because in these new blood vessels, the vessel wall isn't completely sealed. So these endothelial cells don't seal up against them because they're new.
They do eventually close up though, but just at the beginning they don't, okay.
When they're developing, they don't. And the holes that are left in the blood vessels are about the size of the nanomaterials. And so a nanomaterial can go through that that hole and be retained preferentially near the tumor itself.
Okay, so there's a little tiny arrow there now that you can see that shows.
You can back that up and do that again. So if we imagined a nanoparticle was in the blood vessel and it's traversing along with the blood, and eventually it will encounter one of those holes and go through it with some probability. And that we can exploit to deliver nanoparticles to tumors, either in a therapeutic sense or in a diagnostic sense. And so what a nanomaterials person would do is try and design that particle for the best efficiency of that process. And that's really what this study is all about. How do we understand the properties of the nanomaterials to best get them close to and even within the tumor itself? And so if we move along from that idea to imagining a kind of a diagnostic test, so this is the so-called nanoparticle cocktail, and at this point it's a hypothesis, more so than something that people can actually take to try and diagnose their cancer. But if you imagine a series of different kinds of nanoparticles that would be attracted to or retained in tumors, and you could administer that mixture to a patient, circulates around within them. So in the middle of this is meant to be a blood vessel that maybe has some tumor tissue around it. And then a different mixture comes out the other end once it's passed by the tumor. And if you can analyze that mixture in some ways to notice that maybe different colors of particles come out or different sizes, then you can use that as an indirect diagnosis of the cancer tumor itself.
Because the ones, you can sort of get the story by the ones that stayed behind versus the ones that came out the other side.
Yeah, exactly. You've designed the mixture so that when the mixture changes between going in and coming out, that allows you to diagnose or have a preliminary diagnosis that a tumor might exist. And the beauty of this sort of thing would be is detecting tumors that are so tiny that they would never come up on an MRI, for example. All right, so how do we go about testing this hypothesis to see if it would actually work? So we need to look at models of cancer. Certainly can't go into a human condition straight away. And so in this slide, what we have is tests that we've done on the blood vessel system of chicken embryos. And so on the panel A, you can see that there's an egg on a microscope stage. There are nanoparticles that get injected into the bloodstream. So slide B is through a green filter. It's not actually green egg, in deference to Dr. Seuss. And what we have, the dark parts are the blood vessels, the big blood vessels with blood flowing through. We've got it labeled as where the vessel wall is. You can see that along the white arrows, there's a little bit on the left-hand side that seems to have cleared out the blood vessel, so it's not as dark. And that's where the injection of the nanoparticles have gone in. And then we measure to see how fast they're leaving the bloodstream and see how that depends on the properties of the nanoparticles themselves, which could be their size or their surface charge or their surface chemistry.
Can you just inject this with a needle into the blood?
Yeah,
okay.
Yeah, exactly. And we have special procedures to fabricate needles small enough to do this. And then the students who work on this actually become microsurgeons in a sense to be able to do this. All the injection is done under a microscope.
Wow.
Yeah, it's really, really interesting stuff. And the first thing that we notice in panel C is if we've injected a certain kind of nanoparticle, in this case they're called quantum dots, that they immediately go to the blood vessel wall. And that's true of all particles that have a certain charge. So if their surface charge is opposite to that of the blood vessel wall, they'll immediately adhere. And it's sort of in a way like when you have two magnets together and they'll line themselves up. So negative and positive charges attract each other electrostatically, and that's a very strong interaction. So what this tells us is that if you want particles to circulate around and find a tumor, they shouldn't be positively charged if it's going to be blood administered. So we moved on to particles that have more of a negative charge so that they can circulate around a little bit longer and maybe find that tumor. And so this is the result of that study that took us a number of years to complete. And so in the top panel is another blood vessel. It's kind of like a slice along the blood vessel. So there's the the blue parts are the blood vessel wall, and the black in between is the center of the blood vessel, also known as the lumen. There's a little bit of pink color on the left-hand side, and that's where some of these particles have started to accumulate. And in fact, we believe are going through the blood vessel wall through these perforations that we talked about before. And so we can measure how fast the particles leave the blood vessel circulation going through these perforations and just see what that depends on. And we might have thought originally that it would depend on the size of the particles, how fast they get to move around, and that's certainly true. The plot in the bottom left-hand corner is us trying to figure out exactly what the mechanism might be. And so this plot is what's known as the rate constant for particles leaving the bloodstream. Also, you can think of it as how fast they go. And on the bottom is plotted against some factor in size, and it's actually 1 over the square of the radius. And so if you think way back to the time you were thinking about geometry and circles and stuff like that, so r squared, pi r squared, that's like the surface area of a circle. So it seems like if the 1 over r squared gets bigger, that means the r is getting smaller, the faster these things are leaving the blood vessel system. So something to do with the surface area of the particles is making them leave the blood vessel faster. And so the hypothesis that we have for that is that it's the actual footprint of the nanoparticles as they traverse over the holes in the blood vessel. And you can think of this if you had a handful of marbles of different size, and perhaps you're rolling them along a putting green or something like that, so you're imagining a hole, and as the particles cross the hole in the putting green, the smaller ones have a way higher probability of dropping into the hole. And it turns out that that probability actually goes like the radius of the particles squared. In a sense, it's the shadow that they cast onto these holes in the blood vessel. So if we wanted to design for certain circulation times, we could simply change the size of a given nanoparticle. And we found in our study that was regardless of what the particle was made of. So this kind of process depends, maybe not exclusively, but primarily on the particle size.
That makes sense if you say the marbles rolling through the hole.
Okay, so let's ramp up the complexity a little bit. So that was a blood vessel. There was no tumors involved, but the chicken embryo blood vessels are so-called angiogenic, so they're newly developing. They have these perforations in them. Now what happens if we do a similar kind of experiment, but do them with an animal model of a tumor, and trying to pay attention particularly into this collagen matrix or collagen fortress that gets built around the tumor. So in the top panel, those 4 kind of yellowish-looking panels, and the collagen around the tumor is stained in pink. So these are tumor cells. It's a slice through a tumor, and we're looking to see how much collagen exists in these different slices. And these different slices can be thought of as different ages of the tumor or stages as it develops. So they're taken off at different growth periods. The tumors are different sizes, and you can see pretty clearly that there's more pink on the left-hand side than there is on the right. And so as the tumor ages, actually there becomes less collagen per tumor mass. So when it's first developing, there's a lot of collagen around, and maybe we can exploit that. What you see in panel B is just a different way. So scientists like to look at things from a bunch of different angles and using a bunch of different techniques. So B is a different technique, basically, to support evidence that collagen changes as a function of tumor development.
Does it get thinner?
Less of it and more on the surface and less throughout the tumor kind of thing, which is, I guess, what the panels along A kind of indicate. So yeah, it becomes more of a shell as the tumor develops. And we also, as scientists, particularly physical chemists, of which I count myself one, like to plot data, just like the last slide to see things. So in slide C, we've got the tumor size on the bottom of the plot, and that can also be thought of as the tumor age as it grows, it gets bigger, and the amount of collagen on the right. So you can see that the total amount of collagen changes, like we were just talking about, and it's an interesting question as to whether the distribution changes in the tumor as well, which it does. And so we're learning more about the tumor itself, and through that, and one could argue only through that, can you effectively treat cancer.
We have a question from one of our participants, maybe getting ahead of ourselves a little bit, but the question is, what makes nanoparticles specific to a tumor? Is it tumor specific? And I'd like to add on to that, do different tumors have different types of collagen?
Right. So the collagen is probably the same tumor to tumor, a little different species to species, organism to organism. The beauty of the nanoparticles is that you can design the surface chemistry on them so they get selectively retained once they can get to the tumor cells themselves. So the tumor cells will be expressing certain molecules on their surface, and you can put a molecule on the nanoparticle, which is known as an antibody. So it's like a lock and key to those molecules that the cell and only the tumor cells are expressing. And so if the nanoparticle goes through the blood vessel system, gets out of the leaky blood vessel, makes its way through the collagen to the tumor cells, it will get stuck on the cells themselves and hopefully actually ingested by the cells, and it's kind of like a Trojan horse then,
right,
and will release the drugs. And that's one of the beauties of nanomedicine is you can hide the chemotherapy inside the nanoparticles, and you don't get the collateral damage of non-specific chemo that folks have gone through because it does go everywhere.
Well, and one of the things I remember you saying before was that the chemotherapy goes and attacks cells that divide rapidly, because that's what cancer does, but the collateral damage there is your hair and your fingernails, et cetera.
Yeah, exactly. Skin to anything where the cells are developing rapidly or turning over rapidly. And so if you can hide stealthily the chemo from those cells by having them selectively retained only in the tumor site, then that should allow both for fewer side effects and less of the drug needed as well. And that could actually reduce the cost. Okay, so for this tumor experiment that we did this in collaboration with a few folks, one of which is the Warren Chan group at the University of Toronto, and he is one of the world experts in designing nanoparticles. And so we used, for this one, gold nanoparticles. There's lots of good reasons for that. They're reasonably bioinactive, so they're not toxic to organisms. You can design molecules on their surfaces just like we were talking about in order to direct them or make them more stealthy. And you can label them with molecules that allow you to see them under a microscope. And so that's what we've got here in panel A. There's a certain polymer on the outside of the gold nanoparticle, polyethylene glycol. The pink stars are meant to be fluorescent molecules so that we can specifically light up these particles when we're looking at a tumor under a microscope. In panels B, we've got electron microscopy images of particles of different sizes. So what we're trying to do here is see the effect only of size on how these particles would interact with the collagen matrix that surrounds a tumor. And so we have a size range in diameter of 15 up to 100 nanometers. And that's right in the size range that we talked about before. Still very, very, very much smaller than a tumor cell itself. In panel C, we've measured the size in solution. The electron microscopy images are actually in a vacuum. And so we need to know what size these particles are once you put them into water or into the bloodstream. And that's what the panel C is telling us, that yes, they do have different sizes. They get a little bit bigger in water because they actually carry a shell of water around with them. And then in panel D, we have a way to measure the charge as well, and that's what the zeta potential represents. So charge, and basically the plots all line up on top of each other, and that says that these particles mostly have the same charge. So whatever differences in their behavior in a tumor environment should be able to be correlated with their differences in size. And that's what we're hoping to test.
Okay.
All right, so now to some results. For everybody out there that loves results, that's what scientists love to see.
Oh, more graphs, look at that.
Indeed. And so the top panel is another slice through a tumor. So again, the pink stains are the cells or the nuclei of the cells. In the middle, there's a pattern that, if you're using your imagination, maybe it looks like some sort of fish, and that's actually where the blood vessel is. And what we've done is administered these nanoparticles to a rodent model for tumors. So it's a breast cancer tumor that's been implanted on breast tissue in a rodent. Inject the nanoparticle mixture into the bloodstream and then follow how far the particles get into the tumor as a function of time and as a function of the tumor development itself. And that's what these plots are trying to suggest here. So the 15, 45, 60, and 100 nm per nanometers, those are different sets of data for the particle of that size. And the different colors in the plots are different tumor sizes, which means different tumor development points. And so you can see that certainly for the blue one, for example, that the shape of that is changing as a function of the nanoparticle size. And so that should give us some indication of how size could give you a diagnosis of the collagen around the tumor and how we might be able to use that to deliver the nanoparticles more effectively to the tumor. A tumor environment is really complex, as is nanoparticles leaving the bloodstream. There's all sorts of components in blood, as anyone that's ever cut themselves can imagine. And so we've tried to peel back the onion to the most basic collagen model that we can. So this is work that's done in Tina Rinker's group here at the University of Calgary. And this is looking just at collagen and how a nanoparticle might go into pure collagen.
Okay, we actually have a question from one of our participants asking us to repeat or remind us the role of the collagen for the tumor. So this might be a good place to ask that question.
Yeah, exactly. So not completely known at this time. Having just started to develop an understanding that the collagen exists, at this point we can speculate. Everything that a tumor seems to do that's different from its original cells seems to help promote and protect the tumor itself. So it's making some sort of barrier, an extratumoral matrix around itself for protection. It's kind of ironic because eventually a tumor will kill its host, and so it protects itself for a certain amount of time, and yet the ultimate fate of the tumor is the same as that of the host. So it's a very interesting question what the role of this collagen is with respect to the tumor. Protection is probably the simplest one that we can imagine, but there might be more complex roles as well.
Thank you.
Yeah, absolutely. So in this follow-up study, we're testing what the parameters are around these nanoparticles strictly with interaction with a collagen matrix. So basically the nanoparticles versus jello. Instead of the nanoparticles leaving the bloodstream, proteins all over the place, jello around a tumor in a living organism. So this should help us understand what might be going on in those more complicated looking data sets on the previous slide. And so what happens here, we've got a solution with nanoparticles in it, in a petri dish basically, and a slab of collagen, and we see how fast the particles go into or onto the collagen. And so this is an animation here showing a typical data run for that. So on the left-hand side of this square plot is where the solution would be with the nanoparticles in it, and the lighter kind of white speckly side is where the collagen is. And these are fluorescence microscopy images over a period of time. And you can see it resets down to where there isn't spikes in the middle. That's basically time zero. And then the spikes in the middle is right where the nanoparticles are accumulating. And it's sure suggestive that the particles accumulate on that surface of the collagen, right at the interface. And in fact, depending on the particle, seem to get stuck there.
And pretty quickly?
Yeah, as fast as diffusion in water will allow them to accumulate. So there's no real strong driving force for this to happen, except when a particle bumps into that collagen interface that it sticks there. So again, this could be some sort of electrostatic interaction like we talked about before, or it could be a specific molecule-to-molecule interaction. And if this collagen model represents what's happening in a tumor, this is really important to know. If the particles get stuck at that interface, that could be fabulous for diagnosing a tumor, but it might be something we need to overcome for treating a tumor, because this interface needs to be got past in order for the nanoparticles to deliver their drugs to the tumor. Okay, so we've got a bunch of complicated results. And so how do we then try to find a way to understand the best mechanism for designing the best nanoparticle for whatever application in cancer we've got? In the absence of trying to do this mathematically, then basically you're always trying to design a new particle and relying almost exclusively on serendipity to understand what's happening. Here we're designing particles of a series of different sizes, potentially different surface chemistries, and seeing what their effectiveness is in getting to the tumor. And that's what this equation is meant to be representing. So a score, this could be a score eventually even that an MD would use to try and decide which particle is best for his or her patient. The importance multiplier is a factor deciding how important the parameter that we're measuring is, and then the beta is the ranking of that parameter. So it could be that speed of leaving the bloodstream, for example. That could be one of the beta parameters, and we decide how important is that for a particular application. For treatment, it could be very important, so that factor might be quite large. Other things, depending on whether this is a diagnosis or a treatment, so we've got here each of these rankings, these factors could be for a question in drug loading capacity. So how much drug could you fit into one of the nanoparticles? That's very important for treatment. The total accumulation at the tumor could be very important as well. Accumulation rate, that's kind of like what we talked about in leaving the bloodstream. And tumor penetration depth, that was the plots that we had before. And that's very important when you're thinking about therapy, but less important when you're thinking about just determining whether the tumor is there or not. So we've done all these experiments, we've mixed and matched all these results to try and get a score and then represent it in some way that makes sense. And there are many out there, including myself, that are visual learners, understanders. So we make heat maps, and that's what panel A is here, for how to figure out the properties of the nanoparticle with respect to what you're trying to do to the tumor. So, for example, on the left-hand side is if it's acting as a therapeutic agent, and then we're looking at the tumor size. So as you go down that left-hand side, the tumor's getting bigger and therefore older, and the nanoparticle size along the top scale. And so red means works very well, and green means not so much. If we're trying to think about a therapeutic agent, it really says that we should be looking at some balance in the smaller nanoparticle range. And that's mostly because these ones can get through the fortress, the collagen fortress, and deliver drugs to the tumor cells themselves. If we're thinking of them as diagnostic agents, and this could be our cocktails, for example, it's not quite the same story. So if we're trying to particularly diagnose early tumors, small tumors, so that's right at the top scale here, these very small ones, then it looks like a particles on the order of 60 nanometers in diameter might be the sweet spot for that. That's the reddest one for that. So that would be a little bit different, and we design a mixture differently based on what we were trying to accomplish. And usually diagnosis comes before therapy, so that would be the first thing we do. In panel B, you kind of see what we'd suggested is a way that you could use this particular set of nanoparticles for both first tumor assessment using the right-hand side heat map, and then later treatment. So once you know what the size and the parameters of the tumor are, then you can get into designing the right therapy.
We have a question that might fit here well. So is the delivery of chemotherapy using nanotechnology likely to work on metastasized cancers, not just a tumor, but once it's moved throughout the body?
Right. So circulating tumor cells are tough for sure, because it's not the same sort of tumor mass. It's not the same anatomy and physiology. So usually once a cancer has metastasized, then the prognosis is quite grim, because it's so hard to treat. These cells are circulating around everywhere. If you had a surface chemistry of a nanoparticle that could identify these circulating tumor cells, then there's some hope. for sure. And so that's one area of research. We're not participating in that area, but that is not impossible.
Some folks are working on that somewhere?
Yeah.
Okay, thank you.
Great. So now we've got the heat map, and so we'd like to do a test of all this, kind of like a blind test. So if we have a particular tumor in an animal model and we're trying to do a diagnosis of it, and we have our particles, what the heat map tells us is if we administer these particles, some of 15 nanometers in diameter versus those that are much bigger, 100 nanometers in diameter, they should accumulate, the smaller ones, about twice as much or twice as fast, depending on how you want to look at it, in a small tumor for an animal model. And so that's the test that we've done to see whether you can actually use this heat map, at least for the rodent model of cancer. And so in panel A is what you see are two images. So these are light microscopy images of a rodent, where the color is the brightest, and the brightest yellow, I guess, almost white. That's where most of the nanoparticles are. And anything that's a little bit red, that's nanoparticle as well. There's a little bit of blue that kind of just helps us identify the rodent itself. And so the tumor is down in the haunch area. On the left-hand panel, you can see a circle around where the tumor is. And shortly after administration, the small particles for sure have gathered, mostly around that tumor. Versus in the right-hand panel, they're mostly in the gut, spleen, liver, kidney. That's where they're being collected, and much less so. And if you go to actually analyzing the data in panels B, C, and D, and particular D contrast, which is what you want for identifying a tumor, you can see that the small particles, the 15s, have accumulated at least twice as much, maybe even more than that. So it does suggest that in these small new tumors in mice that we can design pretty effective diagnosis of the tumor itself. And these tumors, I mean, if you paid attention to the scale, they're incredibly small. So they're less than a millimeter in size, some of these things. And a human tumor will be very tiny once it starts to. And we understand that prevention is the best form of therapy for cancer, but not all cancers can be prevented, so early diagnosis, super important.
Kind of smaller the better, right?
Yeah, yeah. So this is where a bit of the serendipity comes in. So I don't know if there's enough people out there that remember Arnie in this kind of kindergarten-y movie, but one of his famous lines is, "It's not a tumor." And so we understand that everything that we've been doing here is not a human tumor, and there's all sorts of complexity when trying to move from animal models of tumors into humans. And part of that is the lack of understanding of the animal models themselves. And one could argue that some of the largest costs to the development of new drugs is in that transition from animal models to human. And that's often because the animal model has some way of not representing the human condition as well as it should. And that doesn't mean that we need to develop better animal models. Maybe it means that a little bit, but maybe it also means that we need to understand the animal models better and not just presume that when a person implants tumor cells on an animal model that it represents very closely a human tumor condition.
So that could be quite a big gap between the science around the animal model and then transferring it to the human. It's
yeah.
big. It's not as easy as it looks on the slide deck.
An enormous gap. And what our technology potentially allows then is the tracking of the staging of tumors in animals and comparing that with those in humans, which could be from biopsies, for example. And so if we're strictly even just trying to understand how the extracellular matrix changes in an animal model versus in human tumors and being able to correlate those better, it may be that the animal models are developing at a much different rate, that their tumors than in humans, or the anatomy and the physiology of the tumor is developing at a different rate. And we don't know that yet. With this technology, we can, and then correlate when to apply the drugs to the tumor in the animal model compared to an analogous situation in humans.
So not only could this technology help eventually down the road, potentially humans with cancer to diagnose or treat, it's also helpful in furthering that science by understanding how animal models work versus humans.
Yeah, and that's probably the most direct and immediate outcome of this research. And we started off trying to understand cancer in humans, but what we're eventually understanding is how different that might be in humans and models of human cancers. And if you can line those up better, then that could be potentially more cost effective as well, because you're using animal models way more efficiently.
Gotcha.
All right, so I'd like to come full circle. We're getting close to the end. So if we have a bunch of different nanoparticles and we know that they preferentially get attracted to tumors or retained in tumor tissues depending on their size, for example, we could administer a mixture of different size nanoparticles to a patient and run the simulation again. And if the mixture comes out in a certain distribution of sizes, that tells us how big the tiny tumor is inside a patient. And we can start imagining doing this on the animal models themselves. And maybe this only ever gets applied to animal models, but in a way that really helps us understand the connection between them and the human cancers.
How far out are we, do you think, where this might eventually, potentially be a treatment for people with cancer?
There are many steps, and basically involved in everything that we've just talked about in transitioning even this sort of technology to humans. There are many drug administration regulations that have to get passed. We have to make sure that there's zero toxicity to patients as well. And so this is what makes the process slow. And so, maybe 10 years to see something of a diagnostic in humans. That's a fair ways off. So it's more likely and actually immediate that this technology can be applied to the animal model side of things. That's not to say that we're giving up on human applications, but we need to go through regulatory bodies, and we have to be really sure that this would work effectively in humans before taking those steps.
Potentially, though, it could have, as you mentioned, huge cost savings and be a pretty efficient treatment if you can spare some of those side effects that chemo brings and just get directly to the tumor. And
yeah, absolutely. And there are some countries that are using now some small nanoparticle delivery for cancer therapy. So there's a cancer therapy using lipid nanoparticles and a drug that's already approved. So that's one way to get past going through years and years of testing if you're using a drug that's already used. Already approved. And so that's being done in Israel and a few other constituencies for the treatment of breast cancer with a fair amount of success. So that could be coming to a constituency near us in the not too distant future.
We've got a few questions here that I just want to get to before we get to the kind of your conclusion here, because people are obviously very curious about this. There's all kinds of good questions here. One, can you design nanoparticles which could sense DNA mutations in the blood? Right.
So that's a great question. I'd love to say yes, but I'll probably say maybe instead. So certainly, if you can design a nanoparticle with the right chemistry to detect a certain sequence of DNA, that's completely doable. Detecting it in the blood is probably the challenging part of that, because there's so many different kinds of proteins floating around in the blood, and there would be a limited amount of DNA in the blood, but it's for sure possible.
Okay. When is nanomedicine treatment still effective? When would it? The question's written, is nanomedicine treatment still effective? But I think it should be, when would nanomedicine treatment still be effective? When would it be too late?
Right. Yeah, certainly when tumors get to a certain size, chemotherapy becomes largely ineffective, except maybe in shrinking the tumor, leading to surgery. And so the tumors that are nasty, slow developing, like colon cancer, which I have a personal connection with through my mom, that took 10 to 15 years for that to develop to a point where it actually affected her and she noticed something different in her body. And at that point, chemo and nanoparticle-related chemo is not going to help. Basically, some sort of surgery is needed to take care of the tumor. So I think the real strong win for this is early diagnosis. At some point, if the tumor is growing to a certain size, then surgery is probably the best, most effective outcome.
But early days, this might eventually be a type of screening for those, like for the ovarian, colon, some of those prostate, some of those slow-growing detect cancers.
That's what you'd hope once we understand the anatomy and the physiology and how certain nanoparticles could be exploited to diagnose those at a much earlier stage. In a science fiction kind of way, you could imagine having your cocktail once a month and your super toilet detects your urine for different distributions of nanoparticles, and basically it gives you a little heads up to go and see your doctor, or gives you the thumbs up that everything's still good.
I don't even want to ask about a stupid toilet, but just some sort of test that will detect this stuff. Like now women can pee on a stick to find out if they're pregnant. That sort of idea might be down the road.
Yeah, make it as simple as possible. A different mixture might have a different color.
Right.
And if it comes out green, you're solid. And if it comes out red, then go see your doctor.
Go see your doctor. A minute ago, you mentioned about these trials in Israel. And someone has asked, it sounded like a drug that has previously been approved does not need to undergo clinical trials again if it's reformulated with a nanoparticle carrier. Is that true in Canada as well?
So I'm not aware in Canada that we have nanoparticle delivery yet for these drugs, but in principle, that would make it faster if it was a pre-approved drug, because then you're not testing the drug and the delivery formulation for it.
How do you inject the nanoparticle with the drug? How do you bring those two together?
Yeah, so you create a mixture of the drug, and so this is one way that it's done. You create a mixture of the drug and the components of the nanoparticles, and chemists have designed those components so that they self-assemble, they come together. And nanoparticles can be designed to have a hollow center, and so basically they scoop up the drug that's in solution as they form what you could think of as shells or balls with hollow centers in them. And then you just need to separate out the nanoparticles from the remaining drugs in solution that they didn't capture, and that's pretty standard technology to do that. And
then you send the teeny tiny balloons of drugs down the blood vessels to go to war.
Yeah.
Exactly. Now, someone has asked again, are all cancers surrounded by collagen? We brought this up a few minutes ago, that some tumors are and others, as they're kind of out there floating through the blood, aren't there yet.
Yeah, so since we're in the early stages of understanding this whole collagen thing, the honest answer is we don't know. For the tumors that we've been looking at, yes, but that's not an exhaustive number of them. But I think that if it's an advantage for one tumor to develop, then I would be surprised if it isn't an advantage for many of the soft tissue tumors.
Now, you mentioned at the beginning that you started down one path, and then you kind of found this other stuff sort of as science goes. Yeah. Oh, look at that. Imagine that. So tell us a little bit more about that initial path.
Yeah, so initially when we were looking at this, the question wasn't about collagen at all. We were interested in designing nanoparticles in a sensible way to try and get them into tumors, and how the size of the nanoparticle would affect that. And when we started to look at that data, we noticed that there was this correlation with tumor size, therefore tumor age. And that seemed a little bit weird, because the thought of tumors prior to that was that they just get bigger, but that they don't particularly change anatomically other than recruiting some of these blood vessels to them. And so the data was telling us something about the tumors' increase in size and age that was changing something about the way the nanoparticles were interacting with them. And so through discussions with each other and with medical doctors, we started to imagine, is there something around the tumor itself that's changing? The fact that the collagen existed has been known for a while now, but the fact that it's actually changing as the tumor develops wasn't particularly well understood. And anything that's changing gives you a potential handle for treatment and diagnosis. And so that's what we weren't looking at collagen at all when we started this, and yet that becomes the center of the study in the end.
How long have you been working on this stuff, David?
Probably at least 10 years now. It actually started off as a nanoparticle toxicity study, and we're still working on that to a degree. But everything that we were learning about toxicity was also applicable to medical therapy, and that's the you can't really have one without the other. You have to test for tox if you're ever going to use these particles in a clinical setting, but we soon understood that these studies were presenting us with data that could help both on the therapy side and in understanding toxicology.
Okay. Someone is asking, can nanoparticles cross the blood-brain barrier for brain tumors?
Yeah, blood-brain barrier. That's certainly part of what makes brain tumors really hard to treat. So sometimes yes, sometimes no. When the blood-brain barrier is intact, it's hard for particles of this size to cross it. And so it might be hard for this application in a small tumor in the brain. Once the tumor gets big enough, then it does this blood vessel recruiting trick again, and that compromises the blood-brain barrier. And so under those circumstances, nanoparticles can be used to either further diagnose, outline where the tumor is, or as a therapy. And it could be important as a way to mark out the tumor for the brain surgeon to know where the tumor cells are. So if these particles light up in a particular way, and then when they're doing surgery, they're trying to minimize removing healthy tissue, for example.
What would a human treatment plan look like? Someone's asking, traditionally patients go and sit in a chair for several months while receiving chemotherapy or radiation. What would administering drugs to tumors using nanoparticles, how would that be different? Or do we know yet?
Yeah, we don't exactly know yet, except that if it's kind of the traditional IV treatment, that it would be a lot shorter. Part of the challenge with chemo is to get the buildup in the tumor enough so that it's actually destroying the tumor. And because most chemo is non-specific, that takes longer to do. And so the delivering more drugs more specifically to the tumor would shorten the amount of time a person would be sitting in the chair with the IV drip.
Yeah, because nanoparticles would be snipers and chemotherapy is more like buckshot.
Yeah.
Yeah, okay. In the broader sense of nanomedicine, someone's asking if there's a specific problem or challenge that's slowing the advancement of the research.
I would say the lack of understanding of the disease, or maybe the lack of accepting that cancer isn't a single disease. And it's been talked about as cancer, but even though it's genetic in origin, different cancers are wildly different. And so it's maybe smarter to think about breast cancer, think about ovarian cancer as quite different from each other, and that they develop and behave differently, and the chemistries around them, might be a little bit different as well. So I think that in chemistry, we can design nanoparticles basically at will now. We can put any sort of molecule on them. We can make them so that they pop when they get inside a cell and release their drugs. All that's fairly well established. The hang-up point is getting them to the cells in a real tumor, and that has got to come from understanding what the tumor is like.
Do we have much sense that there'd be risk of damage to kidneys with nanoparticles in the system?
Right. So the unlikely any more so than the payload that they're carrying. Nanoparticle formulations tend to be less toxic than the things that they were made of. So there could be some immune response to certain nanoparticles, which would have them accumulate in the kidneys or liver a little bit more, and that's why you do animal testing to know for sure whether that would be happening or not.
Wow, lots of good work done and lots of good work yet to do, I suppose, hey?
Absolutely. Yeah, so some takeaways for folks as we're wrapping up here. So we've talked a lot about the fortress, the matrix around the tumor, and understanding that I believe is going to lead to better prognosis for patients eventually, and maybe sooner than later if we're making the connection with animals. And it was actually through nanotechnology that we discovered this, and so nanotechnology has a real intimate relationship with this. The third point I just stated in a different sense, so I won't belabor that. And ultimately, the less stuff you can use and the better treatment for cancer, that actually lowers the cost, both literally the dollars and the cost in human suffering, and suffering through a chemotherapy, for example. So here's some acknowledgements. My collaborators, Tina Rinker and Sarah Childs here at the U of C. Tina's in biomedical engineering, and Sarah's over in the faculty of medicine. Fabulous collaborators, and Warren Chan at the University of Toronto. He's in biomedical engineering over there. We've had lots of support from various funding agencies for this. The Natural Sciences and Engineering Research Council of Canada, Canadian Institutes of Health Research, and Alberta Innovates have all provided lots of great funding for this project over the last 10 years or so. And the bulk of that money goes into the grad students, postdocs, and undergraduates that do all the work here. So that's a little picture of my group. We're dreaming of spring yet to come in Calgary, standing underneath a nice tree. And basically, yeah, the funding that comes from folks' taxes out there goes into the development of these fabulous students. And without them, none of this is possible. The profs that talk about this have crazy ideas, and these students help us determine how crazy those ideas are.
Well, thank you to your students, to those funders, your collaborators, and thank you to you for coming in today and telling us all about your work in nanoparticles. Really interesting. Thank you, David Cramb.
Been a pleasure. Thanks, Jennifer.
And thank you out there for all your excellent questions. My goodness, quite a few mind-challenging questions there for sure. Sorry we didn't get to all of your questions. Hopefully those resources will be of help to you on the right. Now, we have more webinars coming up. The next one is about clinical depression, what you need to know. That's April 13th. And April 18th, join us for the microbiome, why a few trillion germs can be a very good thing. We also have other, lots of other information at ucalgary.ca/explore collections. As you can see there's the URL. There's all kinds of good information there. There's ebooks, there's videos, there's all kinds of information about our scholars' research. Now, we were hoping that you will take a minute before you leave us today to just answer a survey question about our webinars. You can find it again to the side of your screen, or if it's not there, or you want to like an icon, there's an icon at the bottom of your screen that will bring you to the survey question. We'd really appreciate your help there. And if you have ideas for other UCalgary webinar topics, we'd appreciate those as well. So just give us an email there at that address and let us know what you would like to learn more about, because we certainly have lots of information to share here from the University of Calgary. Thanks again to all of you for joining us today. My name is Jennifer Alford, and we'll see you next time.